Hello. So I will talk about transforming text about neuroimaging into brain maps. So we want to do this because for many neuroimaging studies, as you know, all that is published is a text report. So researchers and practitioners get brain scans of subjects and patients, and then write a report about it where they give the locations of their observations in terms of anatomical brain regions. But in the majority of cases, we only get the text report and not the imaging data itself. And so the structured spatial information that was contained in the images is degraded to natural language. But since there are hundreds of thousands of these journal articles, patient health records, and case reports, we would still like to be able to extract this information and use it for statistical analysis and meta-analysis. So our objective is, given a text, to map it back into the original continuous space and translate it into a brain map so that we can then leverage large corpora of unstructured text and ask questions like which brain regions are relevant for studying Huntington's disease. So first of all, we could think of some simple heuristics. For example, I could take an atlas and look for the labels of this atlas in the text and base my prediction on this. As we see here on an example, this doesn't work. This doesn't work well on the bottom image because people use terms that are not the exact labels of any atlas, and the interactions between terms are important, and even atlases sometimes disagree. So we decided to take a supervised learning approach. In order to train a supervised model, we would need many examples, many pairs of text, and an accompanying brain map. But as I said, we don't have enough of these. So the next best thing are peak activation coordinates. So we automatically extract these from many journal articles in the same way that Neuroscience does. So we have collected around 14,000 full-text journal articles. From each of these, we extract the coordinates, then convolve them with some kernel, which can be a Gaussian kernel or the regions of an atlas. And like this, we obtain a brain map that we associate with the study. And then we also extract the text, vectorize it, for example, by counting word frequencies, and this gives us a row in our feature matrix. And then we simply fit a linear regression to predict the brain map given the vector representation of the text. More precisely, we model these reported coordinates as realizations of a random variable that takes its values in R3, and we try to predict its probability density. So we minimize the mean absolute error because in a finite probability space, this amounts to minimizing the total variation between the predicted and the true distributions. And now I will show you some results. So first of all, we need to check that we can actually predict this probability density, at least better than chance. And since we're predicting a probability density, a natural metric is the average log likelihood of coordinates reported in left-out articles. So this is our metric, and we perform shuffle split cross validation with 100 split and keeping every time around 1,400 articles in the test set. And like this, we can compare different models. So first, we can compare different ways of discretizing the brain, of cutting it into atlas regions or into voxels. And then for each discretization, we can compare different models. And here, in each block, the second box is a very naive sanity check baseline. It's just repeating the mean of the training set. The first box is the heuristic that I talked about of looking for atlas labels in the text. So we can see that it performs poorly, as I explained. And then the last two boxes are performing least squares regression, and the method we propose, least deviations regressions. So the best predictions are obtained by using a voxel-wise encoding and least deviations regression. We can also compare this to what we get by using the maps associated to terms by Neuroscience. And we see that Neuroscience performs significantly better than the baseline, but that the proposed methods perform better. And now we can, once we have trained one of these models, we can inspect it and look at the coefficients that we have learned. Because in our feature matrix, every column corresponds to a word, and we predict something that has the dimension of a brain map. So in the coefficients that we fit, every row is the brain map that the model has learned to associate with a term in our vocabulary. And if we look at these coefficients, they look like what we would expect. So here are the coefficients for anterior cingulate, left amygdala, and right amygdala. And for more functional terms, for object recognition, we see the fusiform gyrus, and for speech, we see the auditory cortex. And I mentioned that this enabled us to pull together text-only studies. So as an example, imagine that I want to discover which brain areas are associated to Huntington's disease in the literature. This disease is not mentioned very often in the corpus that we have collected, for which we have coordinates. So we don't have enough examples of articles about Huntington's disease along with coordinates or brain maps to perform traditional meta-analysis and ALE. But what we can do is fit one of the models I talked about on this small corpus of 14,000 labeled articles, then collect a much larger corpus of 140,000 articles for which we only have text and not coordinates or brain maps, encode this larger corpus, and then contrast the maps we obtained for articles which mention Huntington's disease against the mean distribution. And like this, we obtain this contrast in which we see the putamen and the coded nucleus. And we can repeat this experiment with other diseases. And here is another example for Parkinson's disease. And we also see the brainstem and the motor cortex. So to conclude, we have built a framework for translating text into brain maps and spatial probability distributions over the brain. We have implemented a quantitative validation setting, which allows us to see that using voxel-wise encoding is better than using atlas regions for this task, and that least deviations regression outperforms least squared. And we have seen that this framework allows us to pull together a large number of documents for which we only have text and not brain maps or coordinates, and perform meta-analysis and statistics on their findings in brain space, and therefore leverage the spatial information that they contained, which was very hard to extract before. So please come and talk about this at the poster 2539. And now I would like to thank my advisors and the parietal team at INRIA.